Welcome and thank you for standing by. At this time, all participants are in listen-only mode. During the Q&A session, if you'd like to ask a question, you may press star 1 on your phone. Today's call is being recorded. If you have any objections, you may disconnect at this time. And I'd like to turn the call over to Gina Anderson. Thank you. You may begin.
Good afternoon. I'm Gina Anderson from NASA's Office of Communications. Thank you for joining us today to talk with experts about science investigations launching on SpaceX's 25th commercial resupply mission for the agency to the International Space Station. SpaceX is targeting next week for the launch of its Dragon spacecraft on a Falcon 9 rocket from Launch Complex 39A at NASA's Kennedy Space Center in Florida. Here to talk with us today about some of the research launching aboard the SpaceX Dragon are Dr. Kurt Costello, Chief Scientist for the International Space Station Program and Research Integration Office Deputy Manager, who will discuss the breadth of research being done aboard the orbiting laboratory. Dr. Robert Green, JPL Senior Research Scientist and Principal Investigator for the Earth Science Mineral Dust Source Investigation, or EMIT. James Wall and Jocelyn Tai, team co-leads of the Stanford University team behind the Biopolymer Research for In Situ Capabilities Investigation. Dr. Monica Monici, Principal Investigator for the Suture and Space Experiment. Dr. Sonia Schrepfer, Principal Investigator for the Immunosensescence Study. And Dr. Janet Jansen, Principal Investigator for the Dynamos Experiment. Our speakers will provide a brief overview of their research, and we will open the lines for a few questions after each speaker. If we have time at the end, we can take additional questions. Your phones are on mute now. To get into the question queue, you can press star 1 on your phone at any time. We will call on you, and the operator will open your mic so you can ask your question. We ask that you please stick to one question. If we have time, we'll allow reporters to ask a second question. About 1 hour after we conclude, you can listen to a replay of this teleconference by dialing toll-free 866-407-9261, 866-407-9261, or by visiting the NASA Videos YouTube channel. To start, we'll hear from Dr. Kurt Costello, Chief Scientist for the International Space Station Program and Research Integration Office Deputy Manager. Over to you, Dr. Costello.
Thank you, Gina. At NASA, we're eagerly awaiting the arrival of SpaceX's 25th commercial resupply mission. The International Space Station is an amazing multidisciplinary lab, offering unique access to the long-duration microgravity and the low Earth orbit space environments. To date, we've had over 4,000 researchers perform over 3,000 investigations, resulting in over 2,000 publications from over 100 countries. Each CRS mission brings a wealth of new research to the station. SpaceX missions are specifically helpful in their ability to return samples developed in space to researchers on the ground for further analysis. Because of this, you'll see a significant number of biology and biotechnology investigations on this flight, and we'll be talking to a number of those researchers today. This flight also represents a number of applied science investigations as part of our in-space production and applications initiative. These investigations are designed to test out and refine cases for the productions of goods in the LEO environment for the use, benefit, and potential future sale upon return to Earth. NASA and the ISS National Lab is investing in these initiatives as part of our investment in the commercial LEO marketplace. An example of this is several of these investigations are flying as part of the Space Tango Lab Mission 27. The ISS is also an amazing platform to view and conduct research on the health of our planet at a global scale. We're excited about the arrival of the EMIT instrument which will be installed on our external logistics carrier 1 during the mission. Dr. Robert Green will be available later in this call detailing this investigation in his briefing. With that, I'll turn it back to Gina, and we'll introduce the researchers.
Thank you, Dr. Costello. Now we'll hear from Dr. Robert Green, JPL, Senior Research Scientist and Principal Investigator for the Earth Surface Mineral Dust Source Investigation, or EMIT. Dr. Green is a Senior Research Scientist and Fellow at the JPL, and Director of the JPL Micro Devices Laboratory. For more than 25 years, his research has used advanced imaging spectrometer instrumentation to test hypotheses and pursue scientific investigations and applications research on Earth and for discovery through the solar system. Dr. Green, over to you.
Oh, thank you. Thank you very much. It's a true pleasure to be here and to talk about the exciting science that we're going to pursue with EMIT. So EMIT stands for the Earth Surface Mineral Dust Source Investigation. The acronym says what it does. We call it EMIT for short. First question we ask, what is mineral dust and why is it important? Under conditions of high winds in the arid land regions of our planet, mineral dust is emitted into the Earth's atmosphere. It can travel thousands of miles. In 2020, dust from Africa reached the Caribbean, it reached Florida, it reached Texas. It is a global phenomenon. Earth has a mineral dust cycle. Why is it important? When it leaves the surface, enters the atmosphere, it can absorb and reflect sunlight, heating or cooling our planet. It can form cloud nucleation sites where it plays a role in how clouds are formed and changes atmospheric chemistry in addition. When it's deposited in the oceans, it's a nutrient and can cause algal blooms. When it's deposited in rainforests, for example, in South America, it can be an important nutrient for that ecosystem. When it lands on snow, it can accelerate the melting. This is critical for water resources in many parts of the world that rely on snowmelt for their water supply. And then, of course, for us, it can be a visibility impact, and in fact, it can be toxic and a hazard based on its particle size. So it interacts throughout the Earth system. Right now, our understanding of dust in the atmosphere, we see it with some of the satellites, but we really need to be able to model how dust interacts with the full Earth system. So we're using Earth system models, which model the whole Earth system. One piece of the Earth system is the dust cycle. Right now, that dust cycle is constrained by only 5,000 measurements of mineral samples for the entire planet Earth, arid land regions. EMIT is going to launch to the space station and use spectroscopy to make more than 1 billion observations of the arid land regions and determine the mineral composition with spectroscopy. We will use these new measurements, 1 billion, to update those Earth system models and bring better fidelity to the Earth system models so we understand how mineral dust is interacting with our planet today much better than we do right now. There are big error bars. And once we have this information, we can also run these models into the future to see how things might change under various climate scenarios, 50 and 100 years from now. So EMIT is going to close the gap in our knowledge of what types of minerals are on the surface that get launched into the atmosphere. And we're going to make this important contribution. It's a cost cap mission that we're focused on. I will say there's added value to NASA and our nation in that these measurements can support many other applications. Looking for natural resources, we'll see the spectral signatures of lithium-bearing minerals at the surface. For example, we can see the characteristics of vegetation to understand biodiversity in new ways. There's a host of additional value that will come from our measurement that we'll make available. And lastly, I will say EMIT is a precursor and will provide preparatory information for NASA's Earth System Observatory, which is the next big systematic observatory system being put forward for our planet by NASA. So that's a quick summary of EMIT and its planned contributions. And we're so excited to be here and ready for launch.
That's great, Dr. Green. Thank you. We'll now go to reporters for questions for Dr. Green. Please please press star 1 on your phone to be entered into the queue. Please identify to whom you're addressing your question. In this case, it will be Dr. Green. And Ted, please let us know when we have a reporter in the line.
There is a question in the queue from Jeff Faust with Space News. Your line is now open.
Good afternoon. Curious for Dr. Green, why is the ISS the best platform for this experiment versus, say, a standalone spacecraft that might be able to operate, say, in a sun-synchronous orbit and provide global coverage? Thanks.
So the ISS is very good for our refined, focused objectives for this mission because we're focused on dust sources. Those occur in the air-land regions, and the ISS gets us many opportunities to observe these regions over the course of a year. We'll have repeated observations because there may be some clouds on occasion, and of course, we want good illumination, and there may even be dust in the atmosphere interfering with our understanding of the surface composition, but the ISS offers us so many observations of these regions of our planet in a year. It really is very well suited to this investigation.
Thank you. Do we have any other questions from reporters for Dr. Green?
I'm showing no further questions at this time.
Okay, Dr. Green, we have another question for you from us. How will EMIT help researchers model weather and climate?
That's a great question. Again, the way we understand climate now in the future, we make many observations, NASA, from many platforms. They're all important, but often they're reduced to parameters that go into these big Earth system models that try to give us a handle on this wonderfully complex planet that we live on, so we can understand what's happening today, and then if we've got good evidence that we're doing well today, then we can run them forward to see what might be happening in the future, and that's the core of climate modeling and the IPCC. What underpins all these models is very good initial conditions, as well as very good equations modeling the system. So EMIT is going to help by providing accurate initial conditions as it relates to the surface composition of minerals around the planet. And again, as I said, we're going to go from a state today where those initial conditions are traced to analyses of 5,000 samples, which isn't so many for the entire planet. And we're going to replace those with updates from our direct observations of the surface of the air-land regions and more than 1 billion observations using spectroscopy as the tool to determine what the minerals are for those initial conditions for the Earth system models.
Thank you, Dr. Green. And we will be seeing you at launch next week, so it's exciting. We'll be able to talk to reporters again. Thank you so much.
I'm very much looking forward to it. Thank you so much.
You're welcome. Next, we will hear from James Wall and Jocelyn Tai, team co-leads from the Biopolymer Research for In Situ Capabilities Investigation. Jocelyn Tai is a sophomore studying computer science and political science at Stanford University. Jocelyn is interested in tech policy and involved in the Stanford Student Space Initiative, working on Mars exploration and space policy. James Wall is a sophomore studying mechanical engineering at Stanford University. They're an active member of the Stanford Student Space Initiative, where they work on projects related to Mars exploration, rocket propulsion, and space policy. James, we can start with you and move on to Jocelyn.
Hello, thank you so much for the introduction. I'm going to pass it off to Jocelyn to start with an introduction about the experiment, if that's okay.
Of course, thank you.
Yeah, thank you so much. Hi, everyone. James and I are the co-leads of an experiment to make an alternative to concrete on the ISS. We're working with a protein called bovine serum albumin, or BSA, which, when combined with dirt and water, dries into something about half the strength of Portland cement. When humans explore other planets, they'll need to shield their habitats from radiation. Unfortunately, it's super expensive to transport standard building materials like concrete and steel. So instead, we're going to have to make use of existing resources, like the dirt that's already there. The BSA protein can be combined with almost any dirt-like substance, and the dirt makes up 90% of the mass of the resulting mixture. So this material is a really promising area of research for space construction. This material is being studied at NASA Ames and also by our advisor, Dr. Michael Lepek at Stanford's Bloom Earthquake Engineering Center. Our project team, however, is made up entirely of students from the Stanford Student Space Initiative. Since 2019, we've been working on a prototype of an autonomous brick-making machine that would make use of BSA. However, we did wonder how the bricks might form differently on the Moon or Mars, where gravity is only a fraction of what we have on Earth.
And so we then applied for and were selected as one of 5 student teams to send an experiment to the ISS as part of the Student Payload Opportunity with Citizen Science. During the 30 days our payload is on the ISS, it will create 6 very small bricks, about 7 millimeters across. The payload will operate completely autonomously with no human interaction. The dirt and BSA protein are already mixed together and contained in our brick-shaped molds. And when the payload receives power, pistons will push water through syringes and into the bricks. The bricks then harden as the water evaporates. Sensors record the temperature and humidity, and the whole payload is controlled by a Raspberry Pi Zero. On Earth, evaporation is greatly impacted by buoyancy, causing the water to evaporate upwards out of our bricks. But in the nearly weightless environment on the ISS, the water might evaporate out in all directions. This could change the orientations of the strands of protein that hold together the grains of dirt, which could affect the strength of the bricks. Once the bricks return to us from the ISS, we'll want to know what the structure inside looks like. We'll use an X-ray microscope to take very detailed CT scans of the bricks, allowing us to construct what they look like on the inside in 3D. This is where one of our citizen science opportunities comes into play. We'll be uploading these images publicly and inviting students of all ages to count the number of protein bridges. We're also running control experiments in 6 local middle school classrooms.
This project hasn't been without its fair share of challenges. We encountered all the usual suspects: hardware issues, software issues, even logistical issues. When our payload was selected by NASA in December 2020, we were all working remotely, located all around the world without access to a workspace because of COVID. And so as a result, we had to get really resourceful when we were building the payload. A lot of the hardware is 3D printed, and most of our earlier iterations were built in team members' living rooms. We were very lucky, though, to be able to build our final payload back in the Student Space Initiative's workspace on Stanford's campus.
This project has been an amazing opportunity and learning experience. Many of us are underclassmen, and so we came in with very little knowledge. But over the course of the past couple of years, we've learned many skills, from computer-aided design to coding a Raspberry Pi to laser cutting. We're so grateful to the mentorship we've received from NASA, NanoRacks, and our advisor, Dr. Lepek. Hopefully, over the course of the next year, we'll be able to draw conclusions about the impact of gravity on the formation of this alternative concrete material that will inform future designs of machines that could produce bricks out of lunar or Martian dirt. It's been a long journey, and we're so excited that our payload is going up soon. Gina, we'll hand it back to you.
Awesome. You guys are so smart. Thank you. We'll now take questions for James and Jocelyn. Again, please press *1 to be entered into the queue. Okay, we've got some questions for you. How did you each get started on this interest and path?
I could start with this one. So the two of us, like we said, are part of the Stanford Student Space Initiative, which is one of the largest project organizations on Stanford's campus. The group has a bit over 200 active members, and the two of us are part of our Mars exploration team, which is one of the 7 teams that we have in SSI, the Student Space Initiative. So the Mars team got started with projects in fall 2019, and one of them included this project to design a brick machine that would create these bricks on the Moon or Mars. And so that's personally how I got involved with the project. I was interested in Mars exploration, and this awesome resource was available to us. Jocelyn, what about you?
Yeah, when I got to Stanford, I was mostly just looking for, a nerdy engineering club where I could learn and grow and a place to belong. And so I found the Student Space Initiative and I was looking for teams, and I met James. I was like, wow, James is a super cool person, and this seems like a super cool project. And even though I, didn't really know anything about Mars exploration at the time, I got involved, just started showing up to the meetings, and then, one thing led to another, ended up working on this project, and it's been a great time.
Excellent, thank you. And it sounds like we have a media question for you.
Yes, there's a question in the queue from David Curley with the Discovery Channel. Your line is open.
Thank you very much. This may be unfair, guys, and maybe the NASA folks can jump in. There have been some experiments on regolith and adding other materials to form a structure. Does anybody know the differences between your experiments and the ones that have been up there already, and how this may push research farther? Thank you.
Yeah, great question. So one of the unique things about this BSA protein and the bricks made from this is that the grains of dirt are entirely held together with intermolecular forces, meaning that there's no chemical reaction occurring, and also there is no addition of heat or pressure in order to form the bricks. All you need to do is mix the two materials together and then hydrate them and have the water evaporate. And so because of this, we can make the bricks entirely out of the dirt and the protein. And if we were doing this on Mars, we could hopefully recapture the water as it evaporates. So I don't know a lot about the other experiments that you're referring to. I do know about experiments that have worked with what's in urine, and I've heard of experiments that work with high pressure and high heat. And so we think that specifically this BSA is a promising way to make bricks that doesn't require the input of those heat or pressure or a chemical reaction.
Thank you, James.
Thanks.
Oh, is that Dr. Costello?
No, it was David Curley saying thank you. Thank you.
Oh, cool. Great. Thank you. Okay. Thanks, James and Jocelyn. Joining us next is Dr. Monica Minichi, Principal Investigator for the Suture in Space Investigation. Monica is the Scientific Research Manager of the ASA Campus Joint Laboratory at the University of Florence in Italy. Her expertise is in space and gravitational biology, photobiology, cell biology, wound healing, tissue repair and regeneration, fluorescence microscopy. She's currently the Principal Investigator for the following projects: Suture in Space, ESA Map Whisper, ESA CMSA Grows, and Exposome Signature Neptune. That's quite a lot, Dr. Minichi. Over to you.
Thank you. Thank you also for inviting me to participate. The Suture in Space experiment aims to study the behavior and healing of ex vivo sutured wound models aboard the International Space Station, that is, in microgravity conditions. The sutured wound models are prepared from human skin and blood vessel biopsies derived from plastics and vascular surgery in healthy subjects. Why to study wound healing in space? For two reasons. Because previous experiments on cell cultures and animal models showed that wound closure is delayed in microgravity conditions, and because in future long-term space exploration missions, the risk for injuries, traumatic events, and unexpected emergency surgery could increase. And since evacuation time from space to Earth might be very long, the need for implementing trauma care and surgery in space increases, and wound healing should be regarded as a major problem to be investigated, since it is critical for crew survival. Our skin and blood vessel models will be wounded and sutured and then sent on the International Space Station. And aboard the International Space Station, the healing of the wounds will be studied. Obviously, at the end of the experiment, the samples will come back to Earth for further analysis.
Thank you. Thank you. We will now take questions for Dr. Minichi. Again, please press star 1 to be entered into the queue each time. Okay, Ms. Monica, I have a few questions for you, if you don't mind. Why can some animals completely regenerate parts of their bodies, but humans cannot?
Oh, this is a very big problem of biology. All of us know that there are some animals which are able to regenerate, to perfectly and completely regenerate parts of their body, while humans and mammals in general are not able to do this. Our wounds heal with scars, and this could be an health problem, and we have several diseases due to impairment in the healing process, which range from chronic ulcers to scarring and fibrosis. So we expect that if we better understand some steps, some phases of the healing process, and if we know what are the mechanisms which allow perfect regeneration in some animals and does not allow perfect regeneration in humans, we could make many progress in wound care. So one of the goals of the Suture in Space experiments is also try to analyze some mechanisms at the basis of wound healing in space, and this could also help understand some of these mechanisms which shift the programs, the biological programs, from perfect regeneration to imperfect healing.
Thank you very much. That definitely sounds like it would be something we could use back on Earth. Thank you. We'll now hear from, and can you please put your microphone on mute, please? We'll now hear from Dr. Sonia Schrepfer. Dr. Schrepfer is a professor at the University of California, San Francisco, Gladstone UCSF Institute of Genomic Immunology, and is the principal investigator for, and this one I am going to get tongue-tied, immunosenescence investigation. Work by Dr. Schrepfer is at the forefront of immunobiology and paves the way for treatment of a wide range of diseases. With this project, the team is aiming to understand immune aging and its effect on the stem cell niche in humans. The goal is to identify pathways and mechanisms to prevent and even reverse immune aging. Dr. Schrepfer, over to you.
Thank you, Tina. Yeah, as Tina just told you in the introduction, our project is interested in understanding immune aging. So immunosenescence means aged immune cells in the context of our project. And you might wonder why we are interested in immune cell aging and what it even means. So immune cell aging in patients means that the immune system, which is usually our defense mechanism for infections, for example, is aged and can't really help us in or protect us against viral infections or bacterial infections anymore. And an aged immune system is not necessarily correlating with the age of a patient, but rather with the status of the immune system. So, for example, young patients, like in their 30s, for example, could have an aged immune system because they had multiple latent viral infections. CMV, for example, is a clinical example where patients have CMV infections. The virus stays in our body and constantly gives a target to the immune system, stimulates the immune system, and that can induce immune cell aging. And the reason why we don't really like, from a clinical perspective, aged immune cells is that they are in a senescent state, so they are not able to protect us anymore, but they have a profile that they release certain factors into the bloodstream that affect our health. And one example is those immune cells are everywhere in our body. So they are also in contact with our body-owned stem cell niche. And these stem cell niche we need if we have, for example, injuries in the body that might be a bone fracture. Then stem cells called mesenchymal stromal cells are important to heal that fracture. Or when I think about vascular repair, when we have vascular damage that can occur in any patient, we would need endothelial progenitor cells to repair the vascular damage. And those stem cell niches are actually affected by an aged immune system because the aged immune system is creating an environment that impacts those stem cells. So those stem cells are not able to repair our body in the way they should do it. And we have shown that already in our first space flight study. Those studies are they are funded by NIH cases. And in the first part of the project, we had sent tissue chips on SpaceX 16, that was December 2018, to the International Space Station. And we could show that the space environment induces aging of the immune cells. So it gives us a perfect model to study or induce the aging of the immune system. And at the same time, we could see that an aged immune system, in fact, does impact stem cells. And we investigated mesenchymal stromal cells and endothelial progenitor cells, and does impact their function. And they couldn't repair as good wounds, for example, as they should. What is now the new project is we do know that an aged immune system impacts the stem cell niche. We did identify pathways, and we are in the middle of trying to understand how that immune aging occurs in the hope to prevent it. But this project now goes the next step. So now we want to send our tissue chips with immune cells and stem cells to space. We want to reproduce what we have seen before, that spaceflight conditions induce an aged immune system, and that aged immune system is impacting the stem cell behavior and capability to repair. But this time we want to receive the chips back on Earth, because last time they were frozen in space. We could just understand what is happening, but we could not understand if what is happening in space conditions, that whole aging, if that is reversible. So now for the first time, we will get back those tissue chips with the living cells, and we will keep them after they return in our lab with the goal to understand if immune aging is reversible. And why is it important to us? We really want to understand for our patients who do have aged immune systems and who might be impacted in their capability to repair their vessels or their bone fractures. We want to understand if we could reverse that aging, if there's any pathway we could target, if there's any mechanism we could understand to provide long-term therapy for patients with aged immune systems, and not only think about prevention, but the reversal of the overall immune aging. So we are very excited. We are having a great collaboration partner with BioServe, and BioServe provided us tissue chips we needed because immune cells need completely different culture conditions than the stem cells. And BioServe was able to provide us those chips that we can run those important studies and really return this time the chips from space and run all the functional analysis in the lab after they return after approximately 4 to 5 weeks on the ISS.
Thank you, Dr. Schreff.
Thank you. Oh, yes. Thank you.
Oh, go ahead. Yes, thank you. We'll now take questions for Dr. Schrefer. Again, please press star 1 to be entered into the queue. I have a question for you, Dr. Schrefer. Can you describe what tissue chips are and how they benefit this research?
Yes, thank you. So tissue chips are basically small compartments of small chips where we are plating our cells, so they are the home of the cells while the cells are on the International Space Station. And a tissue chip, our tissue chip is designed that the immune cells we are putting on the chip are able to communicate with the stem cells we are also putting on the chip. So tissue chips offer the opportunity to not only being like the home for the cells you send to the International Space Station, but you can design them in a way, for example, for us, that provides the necessary capabilities we need to do our research. So in our case, the stem cells and the immune cells, that they have contact with each other like they would have in a human body. So a tissue chip, in a certain sense, is able to imitate what we have in a human body. The stem cell is communicating with the immune cells.
Excellent. Thank you so much for that description.
Thank you.
Finally, we'll hear from Dr. Janet Jansen, Chief Scientist and Laboratory Fellow at the Pacific Northwest National Lab in Richland, Washington, and Principal Investigator for the Dynamos investigation. Dr. Jansen has more than 35 years of experience in the field of microbial ecology. After obtaining her PhD in microbial ecology at Michigan State University in 1980, she established a successful career in Sweden for 20 years and was professor, chair of environmental microbiology, and vice dean at the Swedish University of Agricultural Sciences prior to moving back to the U.S. in 2007. Jansen was a senior staff scientist at Lawrence Berkeley National Laboratory from 2007 to 2014 and an adjunct professor at the University of California, Berkeley, and the University of Copenhagen, Denmark. She moved to PNNL in 2014 and has coordinated several research projects at PNNL that are focused on the impacts of environmental perturbations such as climate change and the space environment on functions carried out by the soil microbiome. Dr. Jansen.
Thank you. I'm very pleased to be here. Our project is studying soil microorganisms, and soil microbes are the hidden players of life support system on Earth. I know that's a grand statement, but although these organisms carry out extremely important functions for life on our planet, we really do not know much about them. We do know that soil microorganisms carry out essential functions, including cycling of carbon and other nutrients. They degrade pollutants, they support plant growth, And although some soil microbes are pathogens, the vast majority are beneficial. So soil hosts billions of microbial cells that work together to carry out these functions. Recent research in our lab and other labs around the world have revealed that soil microorganisms are impacted by climate change. And these changes in environmental conditions have a big impact on soil microbes and the functions that they carry out. So the question is, if soil microorganisms are impacted by changes in their environment on Earth, how are they influenced by conditions in space? This is important to know because as humans continue to explore space, including long-duration space flight and eventually the lunar surface and Mars, they are going to need to rely on beneficial functions carried out by soil microbes. On Earth, plants form beneficial relationships with soil microbes that help them to grow. These beneficial relationships can be leveraged in the space environment as well. For example, soil microbes can help to make conditions on the lunar surface and Mars more favorable for plant growth. They can also be used to help grow crops on space stations and during long-term space flight. So our research in the DYNAMOS project, and DYNAMOS stands for Dynamics of Microorganisms in Space, aims to study how a natural community of soil microbes behaves in space. Several conditions on the International Space Station are very different from Earth, and this includes microgravity, altered CO2, and radiation. So we aim to perform a replicated experiment on Earth and the ISS to compare the influence of Earth conditions to those on the space station on our natural soil microbes. So the soil community that we are sending into space consists of 8 different bacterial species that were obtained from a local soil field station at Prosser in Washington State. In fact, we are sending the soil from that same location as an environment for our microbes to live in. The soil will be sterilized, so the only living microbes will be the 8 bacterial species that we add. The 8 bacteria have a special capability. They work together in a consortium to decompose chitin, and chitin is one of the most abundant carbon polymers on Earth. Therefore, we have added chitin to the sterile soil for the 8 bacterial species to grow on. The soil with the added microbes will be incubated in parallel experiments on Earth and in space for up to 3 months. Samples will be taken at different time intervals. After the 3-month period, the samples from the space station will be sent back to Earth on a return flight and shipped to our laboratory, which is at the Pacific Northwest National Laboratory in Washington State. Once at our lab, the samples will be intensely analyzed using the latest molecular tools, we call these omics, to determine which species survived and how they interacted in space compared to Earth. And I'm happy to take any questions.
Thank you, Dr. Jansen. Again, please press star 1 to be entered into the queue to ask a question. Ed, I think it looks like we have a question.
Yes, there is a question from Alan Boyle with GeekWire. Your line is now open.
Thank you. I wanted to ask about why chitin was selected. I guess chitin is second most abundant polysaccharide and cellulose is the first most abundant. And what do you expect to see from the experiment? Do you have any expectations about how the material might be altered differently from on Earth? And then also potential applications. You hinted at that it might make, for example, lunar soil or Martian soil more amenable for example, for cultivation. But if you could go through that would be great as well. So sorry about the 3-in-1 question, but wanted to get all that in. Thank you.
Yeah, the first question about why chitin, and yes, you're right, cellulose is the most abundant, chitin is the next most abundant carbon polymer, is that we wanted to have something that required an interacting community to decompose. And this chitin is a complex polymer, and so it's difficult for one organism to degrade it all by itself. We wanted to have a dependency between the community members so that one might do the initial step in degradation, and then another buddy would then carry on the other steps. So we wanted to have this kind of dependency. And then what we anticipate, so all of these microorganisms are genome sequenced, and we can predict what they might be capable of, for example, during degradation of chitin from their genomes. And we have done some preliminary, we did some preliminary experiments on Earth just to see who might be playing well with whom when we add them together, these 8 species together, and who is competing with whom. So we do have some of that information already. We anticipate that, or our hypothesis is that in the space environment, those interactions may be altered because of the space conditions. For example, in microgravity, it may be more difficult for these different species to find each other, just purely physically, to associate and degrade metabolites between members of the community. And your last question was applications. So chitin metabolism, it's, by itself is not something that's directly applicable to the space environment, but the knowledge that we gain about how soil microorganisms are able to interact is extremely important, because consortia of microorganisms are needed to be able to grow crops in space and to, as you alluded to, make a soil habitat more amenable for crop growth. So just using a single strain is probably not going to be as successful as using these interacting communities.
Awesome, thank you. Are there any other questions for Dr. Jansen?
I'm showing no further phone questions at this time.
Okay, great. We'll now take questions, additional questions for any of our speakers. Again, if you would like to ask a question, please press *1 on your phone. And I do believe I may have missed a reporter's question for one of our other groups earlier, so please feel free to use this opportunity to ask that group a question as well. It looks like we have one, right, Ted?
Yes, there is a question from Jeff Faust with Space News. Your line is now open.
Yeah, I have a question for Kurt Costello. I was just curious what the status of ISS utilization in general is now that you have sort of a steady state of 4 USOS crew members. That's sort of the long desired goal. What are you seeing in terms of crew time utilization, and is that still the limiting factor, or are you running into limiting factors in terms of space for experiments or other resources? Thanks.
Thanks, Jeff. That's a great question. Since we've had 4 crew and actually not too long ago, 5 crew on board ISS for the USOS segment at least, we've been flush with crew time. So we've seen our ISS research sponsors respond and utilize all of that time that's available. However, we're not seeing crew time as the limiting factor for space station right now. Instead, the limiting resource that really is hitting us is the space for larger payloads going up and down, and also being able to return a number of large items that we have on space station. So as you can imagine, over time, we build up a large backlog of old equipment on space station. Some researchers want that equipment returned. Others are okay with seeing it trashed as part of the ISS program. So right now, our most limiting factor really is that large bag capacity for either very large experiments or the return of very large pieces of equipment and experiments. Crew time we're doing great right now on, but as always, we'll continue to watch and hope to maintain that that 4 crew capability as long as we can, because it's really been a boon to the research that we complete on ISS.
Excellent. Please press star 1 to get into the queue if you have additional questions for our speakers. Okay, if there are no other questions, we'll wrap up our call. Thank you for your interest in this important science, research, and technology science meeting telethon that and all of the research that's launching aboard the SpaceX CRS-25 mission. You can follow all the research being conducted during the Expedition 67 mission by visiting nasa.gov/station research news or following @ISS_research on Twitter. Stay tuned to our launch blogs for regular updates in advance of the upcoming launch next week. Stay tuned for details regarding our pre-launch news conference broadcast. And as a reminder, you can listen to a replay of this teleconference by dialing 866-407-9261. That number again is 866-407-9261, or by visiting the NASA video channel on YouTube. Thanks for everyone for joining us. This concludes today's telethon.